This week we get to talk about the nervous system. Introduction to it, we'll talk about the nervous system all the way up through the spinal cord and some peripheral nerves, and then next week we're going to talk about the brain and some cranial nerves. So this week the overview is going to be that we break the nervous system into two separate regions. And so there's going to be a good picture here, probably this one right here, where we can see that the central nervous system consists of the brain and the spinal cord, and that's it. The peripheral nervous system, the other region, is going to be everything else. This is going to go to the eyes, to the hands, to the skin, to the organs, to muscles, everything else. To get there, we originate in the central nervous system, or the sensations come from there, and then to travel we use the peripheral nervous system. So they behave differently, and injuries affect them differently, so we keep them categorized as two separate entities. From there, we need to understand that there are different regions of the nervous system, and we need to know the main players of the nervous system. So when we look at the nervous system tissues, the main one is going to be the neuron. If we look at the neuron, we're going to have a nerve cell body. We sometimes call that the soma. And so in the beginning of the semester we learned about cells, and this is a cell, but now we're understanding or starting to learn or broaden our understanding that cells don't take a specific shape. So muscles, those are made out of cells. Those are long and skinny. Here's another long and skinny, but it has some unique characteristics to it. At the beginning by the nerve cell body, we have these little tiny branches coming off of there, and we call those dendrites. Then we follow this long cord down, and we get all the way to the end, and we have some axon terminals. They kind of look like little frog's feet. So those axon terminals are either going to connect onto the dendrite of another one, of another neuron, or they're going to attach to the effector organ. So that could be a muscle, it could be an organ, it could be anything in the body. These are going to come in two different forms, myelinated and unmyelinated. Myelinated means that there is fat tissue wrapped around it. Now, if we're in the peripheral nervous system, then we're going to be wrapping with Schwann cells. If we're in the central nervous system, then we'll be wrapped with fat from oligodendrocytes. And so here this slide shows you the myelinated axon. You can see there's little tiny circles all over this slide. And the way I analyze this is if we were talking electricity, you've got little cables, and then you wrap those cables together, and now you have an extension cord. If we were to attach a bunch of extension cords together, then we would maybe have like a power line or something along those lines. Basically, we're going from smaller to bigger in much the same way that we do with muscles. And then, of course, we have unmyelinated axons. So these ones, they don't have the oligodendrocyte or the Schwann cell around the outside. The oligodendrocyte or Schwann cell, that's for insulation, and it's also for a thing that we call saltatory conduction. So saltatory conduction, that's the propagation of action potentials, and only along myelinated axons. And so we go from one node of Ranvier to the other. And so here is showing, let's see if we get a good picture. I think I clicked the wrong one. Might have to watch the video for it. So we'll leave that alone. Here's this. This is showing the node of Ranvier right there. So when we talk about saltatory conduction, instead of having to run, or that action potential running the entire length of this cord, instead we do it more like you could picture someone building a wall for a flood where we've got sandbags. And so picture each one of these myelin sheaths as a person, and they're going to take and hand that sandbag from one person to the next person to the next person to the next person. And we do that with influx of sodium at the node of Ranvier. So in our analogy, the impulse is going to be the sandbag, and the myelin sheath is going to be a person, and we do this with a boost of energy with sodium at the node of Ranvier. That video will show that quite well, and it's much, much faster than if we were to take that sandbag ourselves and start from the nucleus at the cell body and run all the way to the axon. It's a much more efficient, much faster way of accomplishing the exact same thing. And this is where things like basically neurodegenerative diseases like multiple sclerosis. What that is that myelin sheath, it gets broken down. So here this picture shows us a lot of the support cells, and I'm not going to go through these individually, but go through them and get to appreciate them. We've got the oligodendrocyte, the astrocyte. You can see that astrocyte connecting the neuron to a capillary bed. So it's a support cell. We're bringing in nutrients directly from that capillary. You can see the microglia. You can see the ependymal cells. This is a very, very good picture. I highly encourage you to learn all of the different types of neuroglia, and they're different from the central nervous system to the peripheral nervous system. And then you've got some slides showing what those look like. Neurotransmitters, you'll want to take a look at those. So neurotransmitters, those are going to be the chemicals within the body that we allow, or that we cause these impulses to go. So we've got some that hang out mostly in the central nervous system, and then we have some that hang out mostly in the peripheral nervous system. And so keep those straight. That's how we're going to contract muscles and all of that fun stuff. You'll also want to take a look at the cross-section of a spinal cord. Here's showing the spinal cord there in the middle. You can see that it's basically suspended inside of the spinal canal. And so this spinal cord, if you look in the very middle of that spinal cord, there's a dark portion and surrounded on the outside by a white portion. So we call that white matter and gray matter, and we'll look a little bit more at that. But what I mostly want you to appreciate here is that we've got those spinal roots coming out. So we've got two different roots, one coming off the front, one coming off the back, and we call those dorsal or ventral. Ventral meaning towards the front or towards the nose, and dorsal meaning towards the back or towards the fin. Those two roots, the dorsal root and the ventral root, they come together and they form what we call a spinal nerve. Then that spinal nerve is what we see exiting out of the vertebral column. A little bit of sensory talk. See if they show us dermatomes. So there's a thing called dermatomes that must be coming up. I think they've got a picture of it. Yeah, here we go. So here's dermatomes. And dermatomes, this is the sensation to the skin. So we've mapped this out, and we understand that if somebody has lost sensation to the T10 nerve root, meaning their 10th vertebrae, something is wrong there, maybe a compression fracture. We can take and go with a pinwheel, and we can run that along the umbilical cord where your belly button is. And while doing that, if it is numb and there's no reaction there, then we understand that that part of the body is no longer functioning. Or we can go to T4 in the male, and that would be the nipple. Or C2 would be the ear and the back of the head. We've mapped this out. This is very beneficial. We can know what's going on with the body. This is my number one advocate as to why we don't hardly need an MRI on someone to know what's going on, because we have these maps that are well thought of, and we can understand how people are doing without seeing. We basically know what the MRI is going to tell us. The MRI is just confirmation. Next up is the spinal nerves themselves. So these are the nerves exiting the spinal column, and you'll notice that it's a bit different than what the vertebral column would give you number-wise. So we've got to have 8 cervical spinal nerves here, where we have 7 cervical vertebrae. So we're going to have C1, 2, 3, 4, 5, 6, 7, and 8. Now the way we accomplish this is that right below the skull, or above C1, is where the C1 nerve root would be. So we begin above the vertebrae. C1 through 7 are above the vertebrae. C8 is below the 7th cervical vertebrae. From there down, we remain in that below vertebrae organization. So from C7 on down, we have a nerve root below that vertebrae, all the way down to L5. So the most typical herniations are L5S1 and L4L5. And so what we're saying with the L5S1 is that the nerve root below L5 and above S1, or below L4 and above L5, but we would call it the vertebrae that is above it in the thoracic and lumbar spine. So if we say that the L5 nerve root is compromised, that means the nerve root that is below L5. Whereas if we say the C3 nerve root is compromised, that means the nerve root that is above C3. If we say C8, now we're back to that below. So if we say the C8 nerve root is compromised, now we're talking about the nerve root that is below that vertebrae. All right, that basically is our overview. Next up, we'll do the brain and we'll do the cranial nerves. That will all be next week. We're getting very close to the end, and then we'll be ready for that midterm. That midterm will be the final. That final will be the exact same format as the midterm. There will be dissection involved. There will be multiple choice involved. So you'll want to continue to work on these sections and practice these quizzes so that when you get to the final, you are familiar and well practiced at removing layers and clicking on specific structures in the specific region that is asked in the questions. If you have any questions, let me know. Otherwise, I'll look forward to talking to you next week.